morning, everyone. My name is Daniel de Oliveira, and I will be the chair of this last technical session of BSB 2021. We are going to have 4 papers in this session, 2 full papers and 2 short papers. And similar to the previous sessions, you can send your questions in the YouTube chat, and then I'm going to read to the presenters at the end of the session. We are going to have all presentations, and at the end of the session, the questions will be sent to the presenters, okay? So let's start the session with the first paper. The title is PIMBA, Applying for MetaBarcoding Analysis. It's going to be presented by Renato Oliveira, that is already here. Good morning, Renato.
Good morning, Daniel.
So you can start.
Okay. Thank you, Daniel. Hello, everyone. My name is Renato Oliveira, and I will present the work entitled PIMBA, a pipeline for metabarcoding analysis. I'm a PhD candidate at the Federal University of Minas Gerais, and also I'm a bioinformatician at the Vale Institute of Technology. To perform a metabarcoding analysis, first we need to define a region or an organism of interest. And once we define that, we can perform some sample collection. And usually this is an environmental sample, so it can be a portion of soil, a pool of water, or even the interior of an organism, for example, the human gut. And once we have those samples, we can perform some DNA extraction and library preparation. And in this step, usually we need to define a gene to be amplified. And we have some marker genes that are very well established to identify a group of organisms. And once we have this genetic material, we can perform some sequencing. So we can use some platforms like Illumina, PacBio, MinION, IonTorrent. And this sequencing will generate some FASTQ files. There are some text files with the generic information that we need to perform some bioinformatic analysis. And with this bioinformatic analysis, we can find the taxa that are present in a given sample. And for that, we have 2 strategies. The first is the operational taxonomic unit, the OTU, which is a clustering method in which we define a clustering similarity to form those OTUs. And the other strategy is the amplicon sequencing variants, the ASVs, that works with exact sequences. So we allow only the difference of one nucleotide, for example. And with this technique, we discard some sequencing and PCR errors. Once we have these OTUs and these ASV defined, we can assign a taxon to each one of them by using a reference database. And with this information, we can have at the end some diversity results, such as alpha and beta diversity information. In this presentation, I will focus on these 3 last steps. And of course, there's a lot of available tools that can perform metabarcoding analysis. You might know some of them. And in the same way, we have a lot of available databases, reference databases. Each one is specialized in one or more single marker gene. And in this table, I summarize all the information, and we can see, for example, that Mothur and Chime are very similar. They can both analyze bacterial data, fungal data sets, and they use the OTU strategy. OBTools can analyze bacterial data sets and any other marker gene that is on GenBank, and it uses the ASV strategy. mBrave can only analyze the marker genes that are present on the BOLD database, and it uses the OTU strategy. And more recently, a pipeline was published, PEMA. It can analyze bacterial, fungal, and animal data sets, and it uses either OTU or ASV strategy. And our intention was to develop a pipeline, which is PIMBA, that can analyze a large range of marker genes and reference databases, and also can use either OTU or ASV. This is the PIMBA workflow. It is divided in 3 main modules, and we decided to implement PIMBA in a containerized way. So each tool and each software in the PIMBA pipeline is present in a Docker image. So you don't need to worry about installing all the softwares and all the tools. The only dependency is that you need to have Docker installed in your computer and the reference database downloaded in your computer, except for the GenBank database, which is a gigantic database, and we implemented PIMBA to work remotely to access this GenBank. And this is in here I show the languages, the technologies that we have in PIMBA. So we have some bash scripts, Python, R, Perl, and the Docker files. The first module is a pre-processing module to perform some quality treatment. It's called PIMBA Prepare. And PIMBA can work with both paired end and single-end reads. In the case of paired end, you just need to inform for PIMBA the forward and the reverse files, the adapters that were used in the sequencing process, the minimum length and the minimum thread value, and then adapter removal will be able to generate some high-quality data. And this high-quality data will be mini-assembled by peer. So peer will take each pair of reads to perform a mini-assemble using another pairs. And there are some other parameters that you can define, for example, the name for the output file and the number of threads that you want to use. This is an example of the command line that you could use with PIMBA Prepare to analyze a paired end dataset. In the case of single-end reads, it gets a little more complicated because when you sequence single-end reads, you can use either single index or dual indexes to multiplex your samples. In the case of single indexes, for example, if you use 4 single indexes, you will be able to sequence 4 samples in a single sequencing run. So in here I have a illustration. Here we have the DNA fragment that we are interested. And here we have the single indexes, each color representing a sample. And here we have the primary sequence that was used. And to analyze this kind of data, you just need to inform for PIMBA Prepare a text file with the barcode index, a FASTA file with these barcodes, and the primary sequence. And then PIMBA Prepare will be able to analyze and correctly label the fragment to each sample. And this is an example of a command line that you could use for that. With dual index, for example, if you use 4 dual indexes, instead of 4 samples, you can pull 16 samples. And this happens because each combination of the indexes will represent a sample in your data. And also, this is, of course, this is a little more complicated, but if you inform all the information, if you inform all the parameters that PIMBA Prepare needs, it will be able to analyze this kind of data as well. And by the time that we were developing PIMBA, we couldn't find a tool that could perform this kind of the multiplexing step. So we included that on PIMBA. And this is an example of the command line that you could use with PIMBA to analyze this kind of data. PIMBA Prepare will generate the high-quality data with a FASTA file, and this FASTA file will be used by PIMBA Run, and PIMBA Run will perform the replication, will discard singletons, and will perform a truncation step. In this truncation step, the streaming step, you need to define the OTU length that you want, and once you define that, we can perform the OTU clustering or the ASV inferring. And this, for that, you need to define if you want to perform OTU or ASV with this parameter. And once you set this parameter, we will have the FASTA file with the OTUs or the ASVs. And with this FASTA file, we can perform some taxonomic assignment. And to perform this taxonomic assignment, if you are using OTU, you need to define the OTU similarity, the clustering similarity, the assignment similarity. If you are working with the GenBank database, you need to define the coverage of the blast hit. And this hit per subject is an interesting functionality of PIMBA, because if you set this as value 1, PIMBA will return the best hit from blast. But otherwise, if you set as 10, for example, values larger than 1, then it will perform a voting system. PIMBA will return the 10 best hits, for example, from blast, and it will perform a voting system to choose a taxon by majority. And this functionality improved our results very, very well. Also, you can define the e-value of the blast. And here you have to pass a database file, a text file, with the location of the databases that you want to use in PIMBA. For example, Silva database, Green Genes database, or even a bold database. And once you have this database, this text file very well defined, you can define a marker gene to be used with PIMBA. And also an interesting functionality is that you can use your own reference database. You just need to prepare it. In our GitHub page, we show how you could prepare your own reference database. For example, with the situation, we could analyze a database of 12S for fishes, ITS for plants, and a database of cyanobacteria, 16S. So this is an interesting functionality. And also with PIMBA, you can perform some curation with Lulu, which is a tool that can discard some erroneous OTUs or ASVs. And you just need to inform PIMBA if you want to run Lulu or not. And PIMBA also will generate some other results, such as the OTU and the ASV table and the taxonomic assignment file, and other other files like the Biome, for example. And with these files, this is an example of a command line that you could use to perform all this analysis. And here we have all the parameters. And with this with these results of PIMBA run, you can you can plot some basic images. For example, if you use the OTU table and the taxonomic assignment file that was generated, and also a file with some metadata, you can use this command line with PIMBA plot and generate some basic plots like taxonomic bar plots, rarefaction curves, and also some biodiversity information with PCOA and MDS. To validate PIMBA, we benchmarked with 3 mock communities datasets. The first dataset was for 16S bacterial, 16S, comprising 20 species in triplicates. The second dataset that we used was for fungal ITS, comprising 19 species. And the last dataset was for metazone cytochrome oxidase, comprising 14 species. The comparison metrics that we used was using true positives, false positives, and false negatives. And with this value, we could calculate the precision and the recall. And with these two values, we could calculate the harmonic mean with using this formula. We have set some parameters as fixed parameters. For example, using the OTU strategy, we fixed the clustering similarity to 97%. And using the ASV strategy, we fixed the the parameter of accepting one nucleotide of difference. A taxon is only considered if it occurs in all replicates except for the COI dataset, which had a less number of reads in one of the replicates. For each dataset, a different range of parameters were used. For example, in the 16S dataset, we varied the strategy, the minimum assignment similarity, the truncation length, and the database that were used. We used Silva and GenBank database. And these are the results, the F1 scores at the genus level. And with this result, we can see that PIMBA got a nice result using the ASV strategy with the Silva database at the minimum assignment similarity of 99% and a truncation length of 200 base pairs. And with this configuration, PIMBA got an F1 score of 98% against 80% of the PEMAs that was the recently published pipeline that PEMA obtained in his benchmark. At the species level, we can see that the best configuration was using the OTU strategy with the GenBank database at similarities of 90, 97, and 99%, but with the truncation length at 250 base pairs. And with this configuration, PIMBA obtained 80% of F1 score against 14% of PEMA. To the fungal dataset, we varied the strategy, the minimum assignment similarity, the truncation length, and also the databases. And for the genus level, we can see that both OTU and ASV using the GenBank database obtain nice results at the truncation length of 100 base pairs and 130 base pairs in every assignment similarity. So with this configuration, we obtained an F1 score of 94% against 86% of PEMA. At the species level, the best configuration was using the OTU strategy with the GenBank database and with the truncation length of 100 base pairs, but in any assignment similarity. So with this configuration, we obtained 74% of F1 score against 26% of PEMA. In the Metazoa COI dataset, we varied the strategy as well, the assignment similarity, the truncation length, and we only used the GenBank database. The results for genus and species level were the same, and we can see here that it was quite homogeneous, and in almost all configurations, PIMBA obtained an F1 score of 100%, which was a very nice result, against 74% of PEMA. To conclude, we show here that PIMBA is a flexible pipeline. You can use either OTU or ASV strategy. You can set different parameters and databases, and you can even use your own reference database. All steps are encapsulated in only 3 command lines using Docker images. And our results showed a high performance when analyzing monkey communities. And we have some good insights. For example, at species level, OTU with GenBank showed better resolution. But ASV also showed better results at the genus level. So the OTU, both OTU and ASVs are strategies that could complement each other. And if you're interested, this is the GitHub page of PIMBA. So you can access it, download it, or if you're having problem running, you can just put there your comment or your suggestions. And I would like to thank my co-authors, Raissa and Gisele, which implemented some of the functionalities of PIMBA. And also I'd like to thank Guilherme Oliveira, which is my PhD advisor, and also the scientific director of the Institute of Technology. I would like to thank the Van Institute of Technology for providing all the infrastructure needed to develop this work. I'd like to thank the Bioinformatics program and the Federal University of Minas Gerais as well, and the organizers of this symposium. So thank you very much.
Thank you, Renato. Very nice presentation.
Thank
And
you.
I will keep the questions for the end of the session. So now I will call the second presenter, that is Fabrício. Fabrício is going to present the paper entitled CellHeap, a workflow for optimizing COVID-19 single-cell RNA-seq data processing in the Santos Dumont supercomputer. So, Fabrício, you can start your talk now.
Thank you very much, Daniel. Can you see my screen?
Yes, perfectly.
Okay. So good morning, everybody. My name is Fabrício Silva. I am a public health researcher at Oswaldo Cruz Foundation. And the title of my talk today will be CellHeap, a workflow for processing COVID-19 single-cell RNA-seq datasets in supercomputers. Okay. I will start my talk with some background information. Then I will present the CellHeap workflow and I will give you some examples of processing results. Then we will have some conclusions and acknowledgements. Okay, so the origin of CellHeap was a systems biology project. A project that investigates the COVID-19 disease under the systems biology approach. The title of the project is The Study of the Molecular Basis of Comorbidities Associated with the Development of Severe COVID-19. And our objective, or our overall objective, is to understand the molecular basis of comorbidities that imply a higher risk of developing severe COVID-19. Of course, using a systems biology approach. Okay, one of the many interesting biological questions that we are investigating in this project is one physiological reaction in humans that's known as cytokine storm. What's a cytokine? A cytokine is a pro-inflammatory signaling molecule that's used by our immune system to fight infections. What's the problem? The problem is that when you have a cytokine storm and the COVID-19 disease triggers a cytokine storm in severely ill patients, is that when you have a sudden huge release of cytokines, you have tissue damage, organ failure, and it may trigger the acute respiratory disease distress syndrome, ARDS. ARDS is one of the major causes of death for COVID-19 patients, for severely ill COVID-19 patients. So our focus here is to understand the evolution of the immune response from mild to severe to the point that you have a cytokine storm throughout the development of the disease, and also to identify therapeutic proposals that will help the treatment for severe patients. That may be, for instance, cellular therapy and some other types of treatment that we are investigating now. Okay. One thing that we have observed regarding COVID-19 is that there was a large amount of papers on COVID-19 that has been published in the last year and in this year as well. So many of those papers, they were based on single-cell RNA-seq analysis. And normally when you publish a paper in Nature and Science, you make the data set available for other researchers to eventually try to investigate other questions. So what we have today is hundreds of terabytes of COVID-19 single-cell RNA-seq data available in public repositories, and we are moving fast to the petabyte scale. So, since we have a lot of single-cell RNA-seq data available, we decided to use this data to investigate the biological question related to the immune system. But for that, we do need a high-performance computing platform to process all this huge amount of single-cell RNA-seq data available from multiple conditions and tissues, and of course, a flexible, configurable, robust processing workflow. This workflow is CellHeap. Okay, for the computing platform, we are using the Santos Dumont Supercomputer. The Santos Dumont Supercomputer is located at the National Laboratory for Scientific Computing, and it has installed capacity of over 5 petaflops per second. It has over 36,000 cores and over 1,100 nodes, so it's a huge machine. And we got access to that machine through a premium account. About our research team, we have a multidisciplinary, multi-institutional research team working in CellHeap and working also on the biological questions of the systems biology project. So we have researchers from Oswaldo Cruz Foundation, the Federal University of Brasilia, the National Laboratory for Scientific Computing, and the State University of Rio de Janeiro. Okay, so what is CellHeap? CellHeap is a pipeline that's composed of 5 phases. The first phase is sample selection and downloading. The second phase is red mapping and expression quantification. The third phase is quality control. In this phase, we eliminate non-viable cells, and we also separate infected and non-infected cells. For the fourth phase, you have the clustering, analysis, and cell type identification. The idea here is to cluster the cells and then to associate each cluster to a cell type using gene markers. And finally, you have the advanced analysis phase. In this phase, you can do, for instance, both gene level and cell level analysis, and this particular phase depends a lot on, there are several algorithms that you can use in the fifth phase, but the choice of algorithms for this particular phase will depend on the biological question that is associated with that particular run of CellHeap. So I will present in a little bit more detail each one of the phases. Okay, for the sample selection criteria, essentially here we are using the same criteria that were defined by the European Bioinformatics Institute, EBI. So for instance, the samples raw data must be available. All series samples should belong to a single species. All samples most come from non-bacterial species, and so on. So, if you have a particular data set, a set of samples that comply with this criteria, we proceed with the downloading. We use the NCBI server for that, NCBI portal, and we have actually 3 choices for downloading. We can use either the NCBI server or the Amazon or Google platforms. But it's something that we're still working on, in the sense that we still have an extended execution time for downloading. So we are now experimenting with some new algorithms to speed up the downloading process. And of course, after downloading, we do sample verification in order to verify if the file was correctly retrieved. For the second phase, the UMI matrix generation phase. For that, we use the cell range count for read mapping and expression quantification. We also use a hybrid genome for read mapping. That's a genome that's human plus SARS-CoV-2. It's a very computing intensive phase. It demands a lot of computing power. And at the end of the phase, there are several quality checks available through cell range count after processing that we should check to see if the execution was okay. So for instance, you have the Q30 measure, you have the level of mapping confidence, and so on. Okay, the third phase is quality control. And for quality control, first we do cell filtering using the CellHeap package. And we have several criteria for cell filtering. For instance, total number of UMI counts, number of genes expressed per cell, and also percentage of mitochondrial DNA. We also do ambient RNA estimation and adjustment in order to adjust the level of expression for each gene. And finally, in this phase, we do the separation of infected and non-infected cells. We analyze separately infected and non-infected cells. And for that, for being able to separate the infected cells, of course, the hybrid genome that we used in the ready mapping phase is fundamental. It's very, very important. Okay, for the fourth phase, we have the cell clustering. First, we do some dimensionality reduction, and for that, we can use a number of algorithms. For instance, you can use PCA, UMAP, Disney, MetaCell, and other algorithms. It depends. We can use sometimes you can use a PCA and UMAP, for instance. First a PCA, then a UMAP processing. And after dimensionality reduction, we do some clustering in order to cluster cells into groups and to associate each cluster with gene markers. Each cluster has some gene markers that will be used for cell type identification. The idea here is to associate a cell type to each cluster. And for that, we can use either automated or non-automated methods, but always with expert curation at the end of the process. And finally, for the fifth phase, we do some advanced analysis on the clusters of cells that were identified in the previous phase. So for instance, we can do either cell level analysis or gene level analysis. Of course, as I said before, the actual algorithms that you're going to use will depend on the biological question associated with that particular run. So for instance, you can do pseudo-time trajectory inference in the cell level analysis, three-dimensional spatial analysis, and other advanced visualization techniques. For the gene level analysis, you can do differential expression, gene set enrichment, and gene network inference. So, for some examples of processing results, for the example of the processing results that I present to you today, we use the bronchoalveolar lavage fluid dataset that's available from NIH geodatabase. What's important here is that this dataset has data from only 12 patients, 3 control, 3 patients with mild symptoms, and 6 patients with severe symptoms. However, you do need tens of terabytes of available storage space for processing. During the red mapping and quantification, there is an expansion of compressed files, and that expansion will require a large amount of storage space available. And we are talking here about only 12 patients. And of course, for this example here, we deployed CellHeap in the Santos Dumont Supercomputing using the Premium Access account. The Premium Access account is the maximum level account available for external users in the Santos Dumont Supercomputer. Okay, so to give you an idea about execution times, for phase 1, using the NCBI as SRA toolkit FastQDump, it's up to 48 hours. It's a long time, and we are working with some kind of parallel downloading in order to reduce that time. For phase 2, we use cell range account. So for cell range account, we need up to 12 hours of processing time per sample in a thin node, in a Santos Dumont thin node, in a B710 compute node. For phase 3, quality control, we use both CellRat and SupX, and normally it takes less than 10 minutes per sample. It's much faster because the input data in this phase is much smaller, for instance, than the input data that you have in phase 2. And finally, you have phase 4 and 5. For that, we used both CellRat and MetaCell package. And normally it takes, for those samples, less than 10 minutes per sample to process. So I'll talk a little bit in more detail here in this presentation about the MetaCell analysis. So what is a MetaCell? A MetaCell is a group of similar single-cell RNA-seq profiles, similar transcriptional profiles that are statistically equivalent to samples derived from the same RNA pool. So, the MetaCell algorithm, it clusters single cells into metacells. And the main argument here is that metacells succeed in capturing most of the biological variation in the data while eliminating most of the sampling noise. So, for instance, here we have a two-dimensional projection of metacells for the patients with mild symptoms, the 3 patients with mild symptoms. So you have here about 66 metacells. The small colored dots are cells, are single cells. And the circles are metacells. And you have a graph and you have edges between metacells. When you have an edge between two metacells, those two metacells are similar in terms of transcriptional profile. And we also did, over this two-dimensional projection, a clustering of metacells. So you have 4 different colors corresponding to 4 different clusters. And for those clusters, we retrieved the marker genes and used those marker genes to do some automatic cell type identification using both the EnrichR and PanGlaudB websites. So you have here an example of systematic cell type identification, automatic cell type identification using hierarchical clustering of metacells. So how will you do this hierarchical clustering? First, you have this confusion matrix that's actually a connection matrix at the bottom of this figure. And then you group metacells into clusters. The blue bars in the top of the figure are clusters of metacells. So you have several clusters. You can see that you have both large bars and small bars, and for each bar you have a set of marker genes. Those marker genes were used to identify cell types, to associate a cluster with cell types. So you can see that are many possibilities of cell type identification. There are several sizes of clusters. You can see that the large bars are associated with broader classifications, and the smaller bars are associated with more strict classification. It includes a smaller number of metacells. So by doing that, in a top-bottom approach, you first get your metacells, and then you do your hierarchical clustering, and then you get your marker genes. You can submit your marker genes to some automatic identification process, and then you can associate clusters of metacells to cell types. And then you can proceed with advanced visualization techniques and other techniques that you need, depending on the biological question that you need to answer. Okay, for the conclusion. I believe that we can state that we are in the era of big data in biology. That means that there is a demand for high performance platforms and tools. Look, I give you an example today of only 12 patients, only one tissue. When you have to analyze hundreds of patients and multiple tissues, I believe that not even the premium account of the Santos Dumont supercomputer will be enough. So we need high performance computing platforms and tools to tackle this challenge. And our answer to that is the CellHeap, which is a general workflow. It's possible to use CellHeap with non-COVID-19 data, with data related to other diseases. We have not done that yet, but we believe that in the future we will try CellHeap with other diseases as well. Up to now, we are only working with COVID-19. And when you work with COVID-19 data to try to answer interesting questions, interesting biological questions related to COVID-19 single-cell RNA-seq data, you do need a lot of data. So there is a demand for a robust computational infrastructure. That's very clear. For COVID-19, it's mandatory. Maybe for other diseases, you can deploy CellHeap on a big server, but for COVID-19, for answering those interesting questions, we do need a lot of resources. Okay, for the acknowledgements, first, I would like to thank the Innova Fiocruz program for funding, for financial support. And I would like to thank also the National Laboratory for Scientific Computing for granting access to the Santos Dumont supercomputing. Without the Santos Dumont supercomputer, we would not move forward. It was simply fundamental for us to have access to Santos Dumont supercomputer, and we thank the National Laboratory for Scientific Computing for granting that access. So, thank you very much for your attention, and I'm available for any questions. Thank you very much.
Thank you, Fabrício. I'll keep the questions for the end of the session. So very nice presentation. Thank you very much. We are going to our third presentation of this session. The title of the paper is FluxPRT, an adaptable and extensible proteomics LIMS. It's going to be presented by Elizabeth Espinosa, who is here too. Hi, Elizabeth. How are you? Good morning.
Good. Good morning.
You can share your screen now, and then I broadcast. Yes, we can see.
Hello, everyone. My name is Elizabeth.
Sorry.
I'm a PhD student in bioinformatics at the Programa de Pós-Graduação em Bioinformática at the Universidade Federal de Minas Gerais in Brazil. And now I'm going to start to present my work called FluxPRT and Adaptable and Extensible Proteomics links. So proteomics analyses are very useful because they may lead to very important scientific discoveries. This thanks to the methods applied for this analysis, but these methods, this data generated for these methods are very difficult to manage and to track. So one of these systems that can aid to this problem is called a Laboratory Information Management System, also called as a LIMS. So in this work, we use the Flux system, that is a workflow-based LIMS, in order to design the FluxPRT proteomics LIMS. Additionally, we present our interactive guide that is focused to help new researchers for proteomics analysis. So we have here the proteomics analysis can have different steps depending to the approach that they are used to. So for example, here we have 3 different approaches, proteomics approaches, like top-down, middle-down, and bottom-up. They are different because they also depend to the biological question that you want to answer. So for example, in top-down, you introduce the complete protein for mass spectrometry analysis, and this is good, for example, if you want to study PTNs, or if you want to study isoforms, protein isoforms. In the middle down approach, there are introduced a big fragments of these proteins because they're passed for a partial enzymatic digestion, and it's also good for some PTNs and some isoforms too. And they'll have a high coverage, half the proteome coverage. In bottom-up approach, we we have an enzymatic digestion step that is complete. So we have introduced the peptides to mass spectrometer analysis. This is good because the majority of the food proteomes that are being studied are using the bottom-up approach. But this lacks in specificity. For example, if you want to study some PTNs or isoforms. So we take in account all this information in order to do our workflow. So we define a workflow like a sequence of activities. Each activity has a set of attributes, or each activity appears here like a box, in a box. These attributes can depend of the type of the data that the user can put in here. For example, we have the date, we have integer, we have a string, etc. One important information is that an activity can have precedence connections. For example, the protein quantification have 3 precedence connections, like sample fractionation, protein extraction, and sample desalinization. So our methodology to do our workflow is we model the workflow using the DrawIO tool. After that, we convert to XPDL workflow using the Wheeling workflow editor in the Flux system. And then we outlet this workflow on the Flux system in order to testing. After very cycle of testing, we did a refinement in collaboration with experts at the Laboratorio de Proteómica y Bioquímica de Proteínas at the Universidad Federal de Viçosa. And finally, we have our workflow that has 14 activities and is enabling experiment tracking and protocol management. So additionally, we have to present our documentation, the FluxPRT specific documentation, but system documentation that you can access here in the bottom, in the help button. And this Flux system is Wheeling using Java technology. The database uses MySQL and Apache Tomcat for web interface. Our documentation, that is FluxPRT specific documentation, this have the descriptions of the activities and you can access that to each activity of the FluxPRT. You click on this and you can access to the interactive guide. So this documentation is built using HTML, CSS, and JavaScript. So pay attention here to the activity tree because the users can register the data according to the approach they use or their approaches they use. For example, we have here 3 examples of flows according to the approach that we apply. For example, top down, middle down, and bottom up. So our system is very adaptable for these approaches, and it's extensible because you can do one experiment for the same activity twice or more times. So we intend to be, the FluxPRT intends to be a separate tool for laboratory coordinations, technicians, and each member of the lab. Their functionalities are sample management, generation of the report in PDF. You can repeat these experiments all times that you need, and you can adjust this process to the different approaches that you're working in your lab. So for future work, we have to adapt our workflow to the NIPI standards. We also need to complete our FluxPRT guide for all the activities, and we also need to have an indexing testing in the proteomics lab, and we also will be translating to English version because actually it has in Portuguese version. So thanks to Capes and Luar for the orientation of the help, and you for pay attention. If you have any questions or are interested in applying this system to your lab, you can contact me. And if you want to test in our system, This is available at this link. This is the username and the password. And this is the link for our interactive guide. Thanks.
Thank you, Elizabeth. I will keep the questions for the end of the session. And now we are going to the last presentation of the session. The author that is going to present the paper couldn't connect, so I will broadcast the video of his presentation. The title of the paper is Metagenomic Insights of the Microbial Community from a Polluted River in Brazil, 2020.
Hi, my name is Carolina. I am from Brazil, and my goal to present my research project about metagenomic insights of the microbial community from a polluted river in Brazil 2020. This is my email. So the drink water in the metropolitan region of Rio de Janeiro, state of Brazil, and some municipalities in the Baixada Fluminense comes from the hydrological basin of the Guandu River, and its potability is guaranteed by the water treatment station of the state water and sewage company, SEDAI, the public company in Rio. Along its route to SEDAI dams, the Guandu River suffers urban influence, being greatly impacted by the receive in natural effluents. Daily monitoring is done to ensure water quality, like taste and odor, presence of cyanotoxins, pH, free residual chlorine, bacteriology. So far, there is no metagenomic work to know what are the other microorganisms that exist in the Guandu River that can cause disease, that are resistant to antibiotics and/or have metals. Metagenomic analysis make this knowledge possible. Thus, metagenomic is the best way to identify the diversity of microorganisms in the environment, especially the more complex ones, as it is a culture-independent technique. So this work aims before metagenomic analysis of the Guandu River to assess its diversity and abundance of disease-causing microorganisms and bacteria. 8 samples of water were collected in the Guandu River in January, February, and March 2022. 6 of groundwater and 2 drink water. After that, genomic DNA was extracted with the nucleus PINKit protocol and used Illumina platform to be sequenced. The sequences obtained were pre-processed with the PRINSEQ software with the quality thread greater than 30. The exploration of the taxonomic and the functional diversity of the microbial communities present in the samples was carried out from results obtained with the BLASTN local alignment program. After sequence binding was performed using diamonds and mapping in the MEGAN program, and all statistical analyses were performed using R program language. So we obtained more than 200 milliseconds from 8 samples collected. Concerning the bacterial domain, 217 clusters were found, and beta-proteobacteria, with an average of 35.51% of, was the most abundant. A total of 3,097 then were found in bacteria. And from these dates, the Shannon, Simpson, and Peale diversity index were calculated. The samples did not show significant difference among the indexes except potable 2, which refers to drink water. So some disease caused general and pollution indicates such as microcysts can induce hepatotoxicity, Escherichia can cause gastrointestinal illness, Enterococcus can cause urinary tract infection, and Prevotella has been linked to several inflammatory diseases. Although we don't know exactly the species of the genera found above, disease results are worrisome and need examined carefully. According to the cluster analysis, the potable 2 sample was the one that stood out the most for being in a certain group, which was expected for being potable water sample, while potato 1 was grouped with the raw water sample, catch 1 here. Furthermore, it was observed that the samples were grouped according to the time collection, which was expected due to the similarity with the abiotic factors. And PC analysis formed two axes of significance that explain 88.7% of the total variation in space, and the first axis explains 65.1% of the total variation, and the second axis explains 23.6% of. The PCA biplot shows three clusters and which variables differentiate the samples. We select some genera that stood out in the PCA analysis for having greater contribution with the samples such as Staphylococcus. The genus includes common cells and pathogens of humans and animals. Some genera cause diverse infections in humans and have become increasingly antibiotic resistant over the past 7 years. Cathonobacter is here, is an important microorganism in the decomposition of organic and carbon in the soil and exhibits significant positive correlations to various arcs, which indicates that this genus is the main potential host for arcs. And here, Rimerella is a genus that has pathological species normally found in birds. So you can conclude that the water analysis shows a high biological diversity, also indicating the presence of pollution and disease-caused genera by sensors. Sustain the reputation of being a polluted river. However, more studies are necessary to confirm the classification of the species. The taxonomic profile was similar among the samples, except for the potable 2 points. This study demonstrates the potential of the methodology in monitoring and surveillance of aquatic environments. As perspective, we will finish the recovery of genomes from the samples to improve the taxonomic resolution and the results. This is our acknowledgement, this is reference, and thank you. Any question, send me an email.
So we are going to start the question answering. part of the session. I'm going to call the presenters again. We have a question here from the audience, but I'm going to start with Renato. Renato, I have a question about your work. You mentioned that you have several bash scripts and Python codes, but the analysis that you showed at the end, you have to collect metadata the entire process, because you have to analyze to generate the graphics and the charts. How do you do that? You have to capture manually, or you have a mechanism to do that?
You are referring to the PIMBA plot step, right? So PIMBA run will generate some result files. For example, the OTU table is generated by PIMBA run. The taxonomic assignment file is also generated by PIMBA run. And this metadata file is a text file that you need to have for any sample that you are analyzing. And you can put some information such as the region of the sample, or the part of the organism of each sample. And this is the only thing that PIMBA cannot automatize, because you have to create this text file, so you can generate some basic plots. And I couldn't mention this in the presentation, but for example, if you have on this metadata file a column with the region of the sample, you can set this column with a parameter in the PIMBA plot command, and all the graphs that PIMBA plot will generate will try to group this column that you have selected in your metadata file.
Very interesting. Thank you. We have a question here for Fabrício from the audience, and Marcelo Reis from Butantan Institute. A question for Fabrício: how a bioinformatician could also get a premium access to Santos Dumont supercomputer?
Okay. Well, you have to write a proposal. The National Laboratory for Scientific Computing has an evaluation board, and you decide on your proposal if you have access to the Santos Dumont supercomputer or not.
I have a question also, Fabrício. You mentioned that you have to need a long time to download data from the repositories, because you have to download the tens of terabytes from the repositories. And did you consider to execute this the second phase? I think the second phase that's compute intensive in the cloud, for example, because the data is already on AWS, and then download the results of the second phase to Santos Dumont?
Yes, we did that. Actually, one of the members of our research team is Alba Mello. She's a professor at the University of Brasilia. She coordinates a research project on AWS. And the problem is that the storage cost for running that on AWS is very high, too expensive. So it's not feasible. So we do need to do the downloading and to process that on the Santos Dumont supercomputer.
Okay. So, Elizabeth, I have a question for you. You mentioned that when you are modeling the workflow, you create that on draw.io and then convert to a XPDL file to upload to your tool. But the XPDL file, you don't have the code to execute. Where do you associate the code to execute for each activity that you define in this file? Is it within your tool or before uploading that to the tool?
After running this workflow, we implement the code in each of the attributes of the activity. For example, to do the cross-linking of the FluxPRT system with the interactive guide.
So you can upload, for example, a Python script or something for each activity that you have defined, right? Okay. We have another question from the audience. Leandro Argolo, I suppose it depends on the size of the dataset, but is PIMBA only recommended for running on clusters or supercomputers, or is it possible to use on laptops or maybe workstation as well? It's for Renato.
So Leandro, the version of PIMBA that is on the GitHub page, I have built to be able to be run in laptops and workstations. In the cluster of the Vale Institute of Technology, we have another version that can use all the processing power of the facility. But if you download the version that is on the GitHub page, you will be able to run in your computer. And as you said, it depends on the size of your data set. And one thing that I have worried was about the GenBank database, which is a very large database. But we have set to PIMBA to work with this database in a remote way. So only in this step, you don't have to worry with downloading a reference database because PIMBA can access it in a remote way. So you could be able to use in your computer, in your laptop.
Thank you, Renato. So we do not have more questions. So I'd like to thank you, Elizabeth, Renato, and Fabrício for your nice presentations and nice work. And I would like you to remember the audience that we have 3 roundtables at 12 a.m. at noon. And you have to register before in Zoom before the roundtables to participate. Do not forget to register. We are going to finish this session now. Thank you for the audience. Thank you all for the presenters, and see you soon.